Hello, my name is Sarah, and today I will be reviewing the study of the mutation of influenza A virus, PAX, decreases pathogenicity in chicken embryos and can increase the yield of reassortant candidate vaccine viruses. In this study, the influenza A virus will be discussed, and it will be heavily focused on the PAX protein and how it causes disease and how it can increase the combination of different strains of influenza to strengthen the vaccine. So a bit of background on this topic. The influenza A virus, as many of you know, is the common flu. The symptoms are runny nose, fever, cough, sore throat, headaches, body aches. There is a yearly vaccine for the flu or influenza. This vaccine is often being studied because of the many different strains of influenza, which is why it creates for so many cases each year. PAX, the protein we'll be focusing on, is a protein that can control an immune response and affects the pathology of the virus. Basically what this means is it is able to control a host and allows it to take control of immune response and increase its ability to fight the flu. And as you can see in the photo, the PAX up in the top corner is entering the host with the mRNA as the host, and is entering the host with the influenza vaccine. In this study, there was two scientific questions. One, can the PAX protein improve the HA yield of vaccine viruses? Proving if the PAX protein is able to stop the virus when it is vaccinated. And question two, can the PAX gene improve antigen yield and decrease the pathogenicity of influenza in embryonated eggs? The materials used in this study were cell lines and plasmids. Cell lines were cultured in DMEM containing fetal bovine serum, penicillin, streptomycin, and geneticin. And they were incubated at 37 degrees Celsius. Antibodies from the rabbit, and these antibodies were polyclonal, so they were derived from multiple rabbits, and a mouse, which is monoclonal and just so derived from just one mouse. And sera, which is a liquid full of proteins that separates when blood is mixed in, and this will allow us to see blood clots. And finally, a site-directed mutagenesis, which and we used a quick change kit, and this just allows us to view cells and antibodies, and primers were used also along with the quick change lighting kit to aid in seeing these antibodies as well. To begin the methods, a protein analysis was done in vitro, and the transcription and translation was done on the rabbit. These were run into a gel, as you can see in figure 3, and the PAX host cell shut off, contrainfecting a line of cells with plasmid with Rhinella luciferase. And Rhinella creates a light in order to act as the genetic reporter. So you can see that in figure 3, the Rhinella was used to be able to see the gels that were run. And also in figure 3, the expressions of the rabbit are shown when the PR8 was injected. And also the activity measured by Promega Glomax 9612 with Promega Rhinella was also taken, and the data in figure 2 is representing all of what we're seeing in the gels on a graph. Continuing on with the methods, reverse genetics rescue of viruses was done, and T93 T cells, which are the embryonic kidneys, were infected with all different strains of the influenza virus. And these were replaced with DMEM. Actually, I'm not sure if I mentioned what DMEM was, but DMEM is a liquid that maintains cells. I know I mentioned it before in a few slides ago. And it maintains the cells so then you can harvest them and, like we did, later infect them in the embryonated hen's eggs. And next we did the RNA extractions, and these were extracted from the hen eggs, and PCRs were performed and purified so sequences could be analyzed in the software, which you can see in figure 6 here. They are, and figure 6 is showing the data of some of the fatality of the hen's eggs and also the data of the eggs that survived and which were monitored daily. And there's also a photo included, D right here, and D is some of the embryonic eggs, the mock embryonic eggs, the ones that did not survive, and the ones that were monitored. And these are just the graphs showing all this data. Finally, for our last method section, we did the chicken embryo pathogenesis model, and the eggs were examined after 10 days and to see if the veins were in the shell or if there was any movement. After the 10 days, the embryos were killed 2 to 3 days post-incubation by chilling just because of the cold air. And the figure shown is showing that the infected eggs and how the infection of the P-R8 virus affected each portion of their body. Shown is the head, body, cerebral hemisphere, liver, and kidneys. And in order to examine these different strains of influenza that had been injected previously, are shown in the WT and the FS columns, and the mock columns are the ones that were not injected with anything. And the tissues were stained and scanned to these. Finally, our results. Most of the data was presented in the previous slides, and the PAX has a low shut-off activity, which lowers activity than other strains of influenza. Question 1 was answered. Question 1 being, can the P-R8 PAX protein improve the HA yield of vaccine viruses? And yes, the P-R8 PAX protein can improve the HA yield of the vaccine virus because of its shut-off activity. The shut-off activity is shown in figures 2 and 3, and these expressions went down significantly once the PAX was injected. In question 2, can the PAX gene improve antigen yield and decrease pathogenicity? The PAX gene cannot improve antigen yield and decrease pathogenicity because of the lack of difference found in each of them, and each of the experiments done. In figure 6, or in figure 5, sorry, there is not much of a difference between the mock slides and the injected slides shown. And in figure 6, the data shown, you can see that the graphs from before it was injected and after it was injected are not very different. This will conclude my review of the PAX protein, and thank you very much.